Thank you very much. Hello, everyone. I'm Vijini, and I'm a postdoc in the FAME lab at Flinders University. I'm going to talk about something totally different now, and it's about a bioinformatics tool I have developed to recover bacteriophages from environmental samples. So, what are bacteriophages? For those of you who don't know, bacteriophages, or phages in short, are viruses that infect bacteria. Uh, Here is how a typical phage would look like. It has a capsid with its genome, a long, or it can be short, tail and tail fibers it uses to attach itself to a bacteria. This transmission electron microscope image shows a bunch of phages infecting a bacteria, replicating inside it, and bursting out while killing the bacteria. So you can see we can find phages wherever bacteria are, and it can be in different environments like lake water, oceans, soil, or even in our human bodies. So in my research, I wanted to develop a method that can recover phage genomes from metagenomic data. We start by collecting samples from our different environments and extract the DNA, sequence the samples, and run them through a pipeline called Hecatome, which was developed in our lab by Mike Roach. Finally, we are left with draft genomes of these different microorganisms in our samples. So how can we get genomes from raw sequencing data? This is where assembly comes into play. So once we have our short reads, the assembly process starts by identifying overlaps between these reads and forms this structure known as the assembly graph. After performing very different simplification steps, we are left with this final assembly graph that has longer sequences called contigs and overlaps between them. Generally, for downstream analysis, we only consider these contig sequences and throw away the assembly graph. Now that we have much longer sequences to work with, I wanted to see which of them came from viruses, or in my case, phages. Many tools have been developed to do viral identification, and some of them are here, CheckV and Seeker. So these tools use individual contigs and run them through a pre-trained model and predict whether they come from a viral genome or not. However, phages can be tricky to assemble, especially from metagenomes, and they often result in fragmented genomes with many contigs. And novel phages also can be overlooked by these tools because they use models trained on known data. So I wanted to see if I can get any additional information from the assembly graph we threw away at the beginning of the analysis to recover complete phage genomes from our data. So this is an example assembly graph. So each of these colored lines represent contigs. And you can see that there are some of these fragments, as well as these connected components that look like a few genomes are like compressed or smushed together. So I wanted to identify such connected components that belong to phages and resolve genomes from them. To do this, I developed a tool called Phables, which can take in an assembly graph and output nice complete phage genomes like this. So let's see what happens inside Phables. Phables starts by identifying what we call phage components that contain conserved phage genes and do not have any bacterial marker genes. And then I take each of these phage components and try to resolve genomes from them. For example, let's consider this phage component with 3 contigs. I start by looking at the sequencing data to make sure that these links are true and calculate that the purple contig has 3 copies, the pink contig has 2 copies, and the brown contig has 5 copies. To make things easy to explain, I'm going to break this component from the brown contig and sort of make it into a linearized component like this. Now I take this linearized component and model it into what we call a flow network. So you can think of a flow network as a system of pipes carrying water. Imagine that each of these pipes can carry a particular amount of water at a given time. For example, this brown pipe can carry 5 liters of water per second, and then it breaks into 2 pipes, and the purple pipe can carry 3 liters per second, and the pink pipe can carry 2 liters per second. So if our system of pipes has no leaks in any of the junctions, how much water would flow through this last brown pipe? Would anyone like to answer? Yes, it's 5. So because water can enter into this brown pipe only from the purple and pink pipes, so whatever the amount flowing through those 2 pipes should pass through. So based on this idea, now I'm going to break this system of pipes into 2 parts that can carry 3 liters per second and 2 liters per second. So this process is done computationally using a technique called flow decomposition. Now I take these 2 parts and backtrack them to our linearized phage component and our original phage component. You can see that these 2 parts actually represent genomes. So here we have 2 genomes. The amount of water passing through the pipes translates to the number of copies each of these genomes have. So this genome has 3 copies, and this genome has 2 copies. So similarly, we repeat this entire process for all of our phage components, and everything has been automated in Phables. Then I benchmark Phables against a recently published viral binning tool called FAM using 4 publicly available datasets from different environments. And I use CheckV to sort of evaluate the viral quality of these genomes. So you can see that Phables has been able to recover the highest number of complete and high-quality genomes combined together from all of these datasets. Then I've put Phables to test on some of the metagenomic samples we have in our lab and was able to find a bunch of novel phage genomes. So out of them, these 2 were quite interesting. So we found them in samples from people with cystic fibrosis and inflammatory bowel disease. So these phages showed no similarity to any of the phages we know so far, and we don't know what they infect. Now that we have the genome sequence to work with, I can actually do more bioinformatics and help our wet lab people isolate these in the lab. So currently, there are less than 27,000 genomes of phages, like complete genomes recovered so far, and Phables can actually help increase this number. Recently, a benchmarking study on methods to recover viral genomes came out, led by Ryan Cook from the Quadram Institute in UK. They used human fecal viromes and counted the number of complete genomes recovered by the mainstream assembly and binning methods shown on the x-axis. And their results found that Phables run on megahit assemblies was the best. So if you study phages from metagenomes or viromes, you can try out Phables. So on that positive note, I would like to conclude my presentation. I've developed a tool called Phables that can recover phages from metagenomic data, and we were able to recover many novel phage genomes. So in future, I would like to add support for long-read assemblies and extend this work to eukaryotic viruses. So here's a big thank you to everyone from my lab and our funding bodies and our collaborators. And if you want to know more details about the methods used in Phables, you can check out our publication on bioinformatics, and this QR code will take you to the GitHub repository. Also, please do check out these awesome posters on phage bioinformatics and metagenomics from our lab. Thank you.